This is looking at one of those fly embryos, taking the entire data set and chopping it in half. And what you may be able to see is a group of cells there in the middle. That group of cells in the middle are the cells that are growing inside as a process of gastrulation. Here's the cells left on the outside. Those cells there are going to become the nervous system. And this is probably the best two-photon microscope anybody's ever built, and it's still not good enough. It's too slow, and as I'll show you in a bit, it has too many distortions. Recently, a huge innovation came out of the EMBL in Germany, a light sheet microscope where instead of shining light through the objective lens into a specimen and then collecting the fluorescence back from the labels, the light is brought in from the side, and in doing so, it gives you the chance to engineer and optimize the light delivery and the light collection. And while this has been really powerful, and in fact it's a technology that's been reinvented over the past century a number of times, what it has the problem of is that the blue light perturbs tissue and it also gets scattered. Now, since we'd been building two-photon microscopes and we were busy cursing at the two-photon microscope for not being adequate, we could take that laser and shoot it in from the side in place of the blue light. And in doing that with a mirror that we wiggle up and down so we get a light sheet that we're generating out of sort of a pencil-like beam of light, what we could get is a very simple modification of the microscope. Here's the microscope objective, there's the camera, here's the laser light coming in through the two sides of the specimen. And when we use this, what we found is that we could get a microscope that was 50-fold faster than our best two-photon and dramatically better at getting the images. So this is looking at the point, the apex of one of these fly embryos. You can see that the individual nuclei are still visible. I should mention that all of these are labeled by the green fluorescent protein being localized to the nucleus. The two-photon microscope turns each one of these round nuclei into something that's elongated about 20-fold, and they run into one another, and it's not possible to image them anymore. So what we want is a technique that no matter where we look in the specimen, we can follow the cells as unique entities. And so here, as we turn the data to the side, you see that you can still follow the individual cells. This turns out to be dramatically less phototoxic. The phototoxicity of the pulsed infrared light is such that it's almost the third power of the intensity that dictates how the cells are harmed by the light. The excitation goes by the second power, so spreading it out over a light sheet gives us a large equivalent of a therapeutic margin, and you can see that the imaging session ends by the embryo hatching and walking away. Well, I guess they don't walk, but whatever it is a maggot does. Okay, so, and this is just comparing our turbo two-photon microscope to the light sheet for imaging through a developing zebrafish brain. And what you can see is that both of them can capture down to single cell level much better than the blue light sheet sort of technology that's been out there. But if we go around and ask quantitatively about the ability to follow all the cells, it's much better in the light sheet than in the laser scanner. For us, the big thing is it's so much faster. So if I wanted to capture an image of the developing head of a living zebrafish embryo, I can do that, but it's going to cost me a half hour. The cells will have been able to move 3 cell diameters in that time, so I'm in trouble. The light sheet does it in 15 seconds. This is actually slower than real time in acquiring the data, good enough that you can watch the heartbeat. And that means that we can then take data from animals where we've made transgenic tagging of the different genes and be able to, in three dimensions, at single cell resolution, be able to follow those cells and do volumetric renderings of these that allow us to really understand where the cells are expressing different genes and how it is that they're interacting. Now, of course, because if I did one animal that was David's shape and one that was mine, there would be slightly different localizations of the gene expression in space. We need techniques to warp the two of us onto the same scale. That's a scary thought again, but I would end up with a hat and a Hawaiian shirt on and shorter, but and with a worse sense of humor. But anyway, I would or more evil sense of humor, I think I should say. But so we've developed the tools based on the tremendous effort that's gone into the Human Brain Project and some of the other mouse genomics efforts. So now we can superimpose the two, and as we do that, we can take what we think are 5 identical specimens, go to align them, and if we do it just by hand, you can see that these 5 different colors or 4 different colors are pretty pathetic. And once we superimpose them, we can get general areas, but the thing that's most important in this is it shows that no embryo is an average embryo. You can see individual cells here that are outside of the preferred territory. In fact, there's a couple cells here that clearly didn't read the program, and they're expressing the wrong gene in the wrong place, or at least by our definition of it. So what it means is that we really need to be able to assess this variance. We need to make atlases of where things express, but to really understand the full developmental program and how it shapes the fates of where the cells go and what they can do, we need to be able to assess those cells that are doing what we think are the wrong things and understand the molecular and cellular dialogues that beat them into shape so we get a consistent embryo developing out of all of them. So we've been doing a number of different molecular tricks to try to harvest that variance and make meaning out of it. And since the individual cells are where the gene regulatory network operates, by looking at what co-varies in the individual cells and between their neighbors, it gives us a chance to eavesdrop on the dialogue between cells and the molecular logic that's within the cell. Now that means we need to ask many different questions of each cell molecularly. We need to be able to ask multiplex questions. And with normal techniques that we use for doing that, like in situ hybridization, we're in trouble then, because we can do a couple labels with a little bit of effort and 3 labels with a tremendous amount of effort. And the problem is that most of the time that we want to see where a target mRNA is, we use a probe, and that probe has some sort of enzyme on it, and that enzyme's used to make some sort of chromogen. And so we end up with these red blobby things within the cytoplasm of the individual cells that are labeled, but they spread more than the limits of that cell. And if I want to do 2 or 3 different genes, I've got to first develop one, then stain the next one and the next, and it's always diminishing returns. So in light of that, a few years ago, the molecular engineers at Caltech, Niles Pierce and his lab, and my lab got together to try to make a multiplex in situ hybridization technique that wasn't as painful. It was based on something that started as a molecular engineering toy system, which were these little hairpins. And those hairpins would like to stick to one another, but in order to do that, they'd have to open up. And so they're blocked energetically when they're in the test tube as these hairpins. And you can see each one has a little star on it. That star is meant to indicate a fluorescent dye. They'll stay there forever in a stable way until they see an initiator sequence. And the initiator sequence binds to this little toe hold, and then it allows the hairpin to open up, because B star here is exactly the sequence it would like to see, and so it's called a strand displacement. It swaps losing one bond for picking up another one. It ends up looking like this, that has this tail hanging off of it that's CB star, which happens to be just the right thing to go to the next hairpin. And so these 2 hairpins will go back and forth once they see the initiator sequence, and each one will provide the initiator for the other. So as a result, these will polymerize up, and it'll space out the dye far enough that we get a linear increase in intensity for a linear increase in the number of dyes. A lot of times when you get too many dyes together, they self-quench, and they can even get darker as they add more dyes. This was the test tube experiment. These are those little hairpins, and we took an extract of zebrafish embryos in this case and poured it on. They don't start the amplification. But if I add that initiator for one pair of hairpins, there's 4 different types of hairpins down here, you can see that the blue ones amplify and come up to this high molecular weight region. The green one or the red one or the yellow